So finally, for this session, I'd like to invite Ben Christensen to tell us about investigating genes of interest in Dupuytren's disease.
All right, thank you for the opportunity to talk here today. I should disclose that you'll notice I'm from the Division of Urology and Department of Surgery, so our primary research interest is actually Peyronie's disease. But since we know that they are likely related diseases with the same underlying mechanism, I think what I'm going to talk about here applies to both equally. So to give a little background, we had previously collected genomic DNA from 16 men that had both Peyronie's and Dupuytren's disease, and for control participants, and used array comparative genomic hybridization to be able to detect genetic variants in those men. What we found were that there were microdeletions in two genes of interest, NEL1 and CDTD-SPL. And you'll see there that the incidence of these two genes, these microdeletions, compared to a genomic database of the normal population, there were statistical excesses of copy number variants in both NEL1 and CTD-SPL. So what are these genes? NEL1 is neuroepithelial-like growth factor 1. It's a growth factor that enhances osteoblastic differentiation and chondrogenesis in the TGF-beta pathway. With a decrease in NEL1, you would also tend to get an overproliferation of fibroblasts. NEL1 has been investigated a little bit previously in animal models, but primarily because of its osteogenic effects. And then CTD-SPL is a small C-terminal protein, also involved in the BMP and TGF-beta pathway signaling. And it has roles in epithelial and mesenchymal transition and myofibroblast differentiation. So both of these genes make sense in the sense of potential pro-fibrotic diseases. We had previously also isolated fibroblasts from Tunica albuginea tissue samples. So that's the tissue that is affected by fibrotic plaques and Peyronie's disease. And then using viral vectors, overexpressed NEL1 in these fibroblasts. And what we found on the panel on the left are going to be pro-fibrotic genes, and then on the panel on the right is anti-fibrotic genes. And we found that with an overexpression of NEL1, we decreased these pro-fibrotic markers and increased anti-fibrotic markers. This suggests that a decrease in NEL1 would have the opposite effect, again, supporting its role in potential pro-fibrotic disease pathways. So what we were interested in doing was looking at downstream effects of regulation of these genes. So we developed gRNA vectors using CRISPR-Cas systems to up and down regulate these genes. For NEL1, we used HEC293 cells because of the baseline expression of NEL1 in these cells. And then for CTD-SPL, we used bladder fibroblasts. Quantitative PCR was used to evaluate the degree of up and down regulation of NEL1 and CDT-SPL expression, and then RNA sequencing was used to perform, was performed to determine downstream effects. Today, I'm only going to present the downstream effects of NEL1 regulation. We're still analyzing the results from CDT-SPL. So we had very robust changes in expression in NEL1. We found that with upregulation, we got a 40-fold increase in NEL1 expression, and with downregulation, we got a 95% decrease in NEL1 expression. This led to a very large number of differentially expressed genes. With upregulation, we got over 10,000 differentially expressed genes, and with downregulation, over 7,000 differentially expressed genes. So that's a lot of genes to try to sort through. So what we did was a pathway analysis to try to determine how these differentially expressed genes actually affected pathways. And a couple things of interest with the upregulation, we saw a decrease in inflammatory process pathways, specifically TNF-alpha signaling via the NF-kappa B pathway. With downregulation of NEL1, we saw an increase in inflammatory pathways, and we also saw a decrease in this G2M checkpoint pathway, That's a checkpoint pathway that's going to be involved in regulating mitosis, and so a decrease in that could increase aberrant fibrosis. So these pathway changes, again, fall in line with what would make sense for a decrease in NAL1 and an increase in profibrotic pathways. And then again, I don't have results to show the analysis of this, but just to show that we've also had success with being able to regulate CDT-SPL, not as robustly as NAL1, but we did see an 86% decrease with our vectors and a 54% increase with upregulation. So in conclusion, we were able to successfully up and down regulate expression of both of these genes. Regulation of the NAL1 expression did lead to large downstream expression profile changes that were related with biological processes that make sense for profibrotic diseases. Analysis of the CDT-SPL is ongoing, and then further work is also looking at ECM deposition from changes in cells that are regulated for both NAL1 and CDT-SPL. I also want to put in a plug here for potential collaboration. We do also have a very robust hand surgery group at the University of Utah and currently have on hand extracted genomic DNA and tissue samples from Dupuytren contractures from over 40 patients. Unfortunately, we don't have funding to do work with those right now, but that would be a great opportunity to pair analysis of whole genome sequencing with both RNA sequencing and epigenetic analysis in those tissues. We may be familiar with the fact that single-cell RNA sequencing in these tissues is very difficult because, as Charlie's mentioned, they're very collagenous in nature, but we have had some success with single nucleotide isolation to be able to do single nucleotide sequencing that way. And I'm happy to take any questions.
Thank you. Any questions? We've got a question. I think that's David at the back.
Just wondering if you know if either of those genes are biomechanically regulated in any way?
Not that we're aware of.
Could I ask, Ben, have you shown expression of either of these genes in the plaques from Peyronie's or in Dupuytren's nodules themselves?
We're working on evaluating the expression profiles in those. As I've mentioned, isolating single cells from those tissues is very difficult. As I mentioned, we've had some success with Dupuytren's contractures to isolate nuclei. We haven't had that success with Peyronie's plaques. I don't know why we're having differences in success rates there, but it's something that we are actively working on.
Thank you. Paul.
Thank you very much. If I understood you well, you use the fibroblasts from the bladder as your cells to work with?
For the CDT-SPL.
Do you think that have influenced the results if you compare it to the Dupuytren's or the Peyronie cases? What's your opinion about that?
Yeah, so we're also working on trying to do this in fibroblasts that are isolated from plaques and contractures. The success rate with those in culture is not high. I mean, basically, you extract the tissue, dice it up, and you hope that something grows. We're also the idea here is to try to get a proof of concept. Even with culture in the fibroblasts from plaques or contractures, there's going to be some influence in growth media conditions as to what their baseline expression profiles would look like. And so we thought we'd start from kind of these primary cell lines and see what the downstream expression profiles look like before we kind of move to that next step.
Great. Thank you very much, Ben.
Thanks.